Hello students, myself is Parminder Singh from Prab Kripa classes. Our today's topic is the fundamental unit of life. In this course, we will discuss why the cell is called the structural and functional unit of life. We will also see what is cell made up of and what is the structural organization of a cell. Further, if the organization of a cell is destroyed due to some physical or chemical influence, what will happen? We will also find out the difference between unicellular and multicellular organisms. Then we will find out what is the shape and size of cells. Then what is the meaning of division of labor in multicellular organisms. Then how do the substances move in and out of the cell? Next, eighth question, what would happen if the plasma membrane ruptures or breaks down? Then why plasma membrane is called a selectively permeable membrane, and what is the meaning of diffusion and osmosis? And another important question, what happens when a plant cell is placed in hypertonic, hypotonic and isotonic solution? Next, what is the meaning of mitosis and meiosis and much more? Discovery of the cell. Robert Hooke was a great scientist. He discovered the cells in 1665. He had designed a microscope himself. You can look at the picture of the microscope he had designed. He looked at the slice of cork through this microscope. What he found after looking at the cork, he discovered that the cork looked like the structure of a honeycomb. That is, the cork was some type relating to the structure of a honeycomb. He found that the cork was made up of a large number of tiny compartments or boxes, and each box was separated from the other by a wall or partition. You can see here that this is the cell structure of the cork, and you can see here that these are the small pores or compartments, and there is a wall in between each of these tiny compartments. He called these tiny compartments or boxes as cells. His observations are recorded in his book Micrographia. Next is cell theory. The credit for developing the cell theory is usually given to two scientists, Sladen and Schwann, while there is also the name of another scientist known as Virchow, who contributed to the cell theory, but he is not as credited for his stuff towards it. Now, there are three basic components of the cell theory. What are those basic components? First is, all living organisms are composed of one or more cells. Second basic component of cell theory is that the cell is the basic unit of life in all living organisms. And the third basic component of cell theory is that all cells arise from pre-existing cells. Now, a question arises, why cell is called the structural and functional unit of life? The word cell is derived from the Latin word cellula, which means a little room. All living organisms can be divided into two types, unicellular organisms and multicellular organisms. In case of unicellular organisms, they are also known as single-celled organisms. That is, a single cell provides the structure for the body, and the single cell performs all the functions that take place inside the body of the organism. All these functions include digestion, excretion, etc. In the case of multicellular organisms, there are multiple cells in the body. And the examples of multicellular organisms are plants, animals, human beings. In the case of multicellular organisms, the number of cells vary from species to species. It is approximated that the human body contains trillions of cells. Sorry. In case of multicellular organisms, cells form the structure of the organism. You can see here that the cells form the structure of the organisms. A bunch of cells working together form the tissue, and a bunch of tissues working together form the organ, and a bunch of organs working together form the organ system, and the organ system further combines to form the organism. The examples of tissue is nervous tissue, example of organ is brain, example of organ system is organ nervous system. The cells in multicellular organisms vary in size and shape depending upon the function they perform. All the functions which take place inside the body of the multicellular organisms, all these functions are performed by the cells. Now the question arises, what are unicellular organisms? A unicellular organism is also known as single-celled organisms. That is, there is only single cell present in a unicellular organism, and all the life processes such as reproduction, feeding, digestion, excretion occur in one cell only. There are some examples of unicellular organisms which are bacteria, protozoa, amoebas, unicellular fungi, etc. Amoeba has no definite shape. It keeps on changing its shape. A white blood cell which is present in the human body is another example of a single cell which can change its shape. So we observe that amoeba can change its shape, and at the same time a white blood cell which is present in the human body can also change its shape. But it does not mean that WBC, that is white blood cell, is also a unicellular organism. White blood cell defends the body against infection and disease. But while WBC is a cell, amoeba is a full-fledged organism which is capable of independent existence. Now, multicellular organisms. You here can see the photographs of beautiful multicellular organisms. These are the four pictures of different multicellular organisms. Multi means many, cellular means cells. It means that there are many cells present in the multicellular organisms. All the species of animals, land plants, and most fungi are multicellular. Now the question arises, how many cells are present in a multicellular organisms? The number of cells which are present in the multicellular organisms may range from a few to many billions. The number of cells being less in smaller organisms does not mean that it affects the functioning of the organisms. All the cells which are present in the multicellular organisms work in coordination with one another for the proper functioning of the organism. It is to be noted that the lifespan of a unicellular organism is short because all the functions inside the body of the unicellular organism, that is excretion, digestion, etc., plays within this single cell. So there is heavy workload on the cell. Due to this reason, the span of a single celled organism is short, whereas on the other side, the lifespan of multicellular organisms is longer because the different cells carry out specific functions. Now we will find out the difference between unicellular and multicellular organisms. First is number of cells. As we have discussed earlier, uni means one, cellular means cells. In case of unicellular organisms, organism consists of a single cell. Whereas in the case of multicellular organisms, as we have already told, there are multiple cells in a multicellular organisms which may range from a few to billions of cells. The next point is size. The size of the cell in the case of unicellular organisms is somewhat larger as compared to the cells in a multicellular organisms. Whereas in the case of multicellular organisms, the size of the cell is smaller. Now, the next point is functions. As the unicellular organism has a single cell in its body, all the functions which are necessary for the functions of the life, such as eating, reproduction, waste removal, moving, etc., are performed by the single cell. Whereas in the case of multicellular organisms, specialized cells perform different life functions. For example, nerve cells. Next is type of role. Cell has only one role, that is as an individual. In the case of multicellular organism, cell has dual role, as an individual and as well as part of the community. Now, next point is workload. Workload in the case of unicellular organisms, workload on the cell is high because all the functions have to be performed by the single cell. But in the case of multiple cellular organisms, there are multiple cells present. So the workload on the individual cell is very less. Next point is division of labor. As there is only single cell present in the unicellular organism, no division of labor is there. Whereas in the case of multicellular organisms, there are a large number of cells present in the organisms and different cells perform different functions. So division of labor is there in case of multicellular organisms. Next point is survival capacity. Death of the cell results into the death of the organism because it is the single cell which is present in the unicellular organisms. Whereas in the case of multicellular organisms, death of a few cells does not result into the death of the organism. Next point is cell replacement. There is no cell replacement, whereas in the case of multicellular organisms, old and weak cells are continuously replaced by the new cells. Examples of unicellular organisms are bacteria, amoeba, yeast, etc. Whereas in the case of multicellular organism, examples are some fungi, animals and plants. Now the topic before us is shape and size of the cells. The cells of different living organisms may differ in shape and size. We have already seen that example of a unicellular organism is amoeba. Amoeba has no definite shape. Its shape may be just like this at one event, and the other time its shape may be this one. It keeps on changing its shape. Within a multicellular organisms, there are a variety of cells. The cells may be blood cells, muscle cells, nerve cells. Some cells are long, other cells are short. Some cells are circular, whereas the other cells are oval shaped. The size and shape of the cells are related to the specific function they perform. A nerve cell is long and branched as it has to transmit signals throughout the body. On the other hand, the muscle cell is small and spindle shaped, which helps in movement. The red blood cells in the case of human beings are spherical. The components of the cell are enclosed in a membrane, which is known as cell membrane. The cell membrane provides shape to the cells of plants and animals. Then in the case of plant cells, there is an additional covering for the cell membrane, which is known as cell wall. Cell wall is present in the case of plant cells. Cell wall provides shape and rigidity to these cells. Now the question arises, what is the size of the cells? The size of the cells in the living organisms may be very, very small, as a micrometer or micron, or may be as large as a few centimeters. The smallest cell is in bacteria, and the largest cell is the egg of an ostrich. The size of the cell has no relation with the size of the body of the animal or plant. It means that we should not think that if the size of the plant or animal is big, the size of the cell present inside it will be very big. The cells in the elephant, it is not necessary that the cells in the elephant be much bigger than those in a rat. The size of the cell is related to its function. For example, nerve cells both in the elephant and rat are long and branched. The nerve cells perform the same function in the elephant and rat, that is the transferring of messages. Now the next point is division of labor in multicellular organisms. There is division of labor in multicellular organisms such as human beings. This means that different parts of the human body perform different functions. A human body has a heart to pump blood. A human body has a stomach to digest food, and so on. Similarly, division of labor is also seen within a single cell. Each cell has got certain specific components. That is, some components are present inside the cell which are known as cell organelles. Each kind of cell organelle performs a specific function which may include making new materials in the cell, whereas some other cell organelle is related to clearing up waste material from the cell, and so on. A cell is able to live and perform its functions because of the cell organelles present inside it. These organelles together constitute the basic unit called the cell. Now, what is the structure of the cell? The word cell has been taken from the Latin word cellula, which means small room. Cell is the basic structural and functional unit of life. If we study a cell under a microscope, we would come across three features almost in every cell. There is a plasma membrane, and there is a nucleus, and there is cytoplasm, which is the fluid present throughout the cell. We can also say that the cell consists of three parts. First is cell membrane, the second is nucleus, and the third is cytoplasm. Within this cytoplasm lies extremely small but distinct structures called organelles, which may be present here and there. Some organelles such as nucleus and Golgi apparatus are usually solitary. Solitary means single. While there are other cell organelles such as mitochondria, chloroplasts, peroxisomes, lysosomes, which can be numerous, that is hundreds to thousands. Now, the question before us is cell organelles. An organelle is a structure found in the cytoplasm that has a specific form and functions. This is the picture of a cell, and there are many cell organelles which you can see are there. The name organelle comes from the idea that these structures are parts of the cell as organs are to the body. It means that as a body has organs, in the same way a cell has small organs which are known as organelles. These are the parts of the cell. Cell organelles are membrane bound structures within a cell. You can see here that these are the cell organelles. These are all cell organelles. These are membrane bound structures within a cell. In prokaryotes, the membrane bound cell organelles and a defined nuclear region are absent. It means that in the case of prokaryotes, membrane bound cell organelles are not present and there is no definite nuclear region. But in the case of eukaryotic cells, they have nuclear membrane as well as membrane enclosed organelles. Some important examples of cell organelles are nucleus, endoplasmic reticulum, Golgi apparatus, mitochondria, vacuole is not appearing as such. Ribosomes, you can see here ribosomes. These are the ribosomes, these small points. The cell organelles are important because they carry out some very crucial functions in a cell. Some of these organelles are visible only with an electron microscope. Large organelles such as nucleus and vacuoles are easily visible with the light microscope. Cell wall, as already told, is present only in the case of animal cells and is not present in the animal cells. It is only present in plant cells. The chloroplasts are also found only in the case of plant cells because they are involved in the making of chlorophyll. Lysosomes is found in the case of, these are the lysosomes. These are found in the case of animal cells. Now, plasma membrane. The plasma membrane is also known as cell membrane or cytoplasmic membrane. It is a biological membrane that surrounds the cytoplasm. This is the cytoplasm all here and there. And cytoplasm is covered by this cell membrane. This is the cell membrane which is of black color. It separates the contents of the cell from outer side, material outside the cell. You can see here that this wall keeps all the cell organelles within the cell. It holds the contents of the cell in place. This cell membrane or plasma membrane is known as is semi-permeable and selectively permeable. It means that it can either let a substance pass through freely, pass through to a limited extent, or not pass through at all. It means that it does not allow each and every type of liquid or gas to freely pass through it. It is semi-permeable. It may allow some gases or liquids to pass through it, or it may not allow at all, or it may allow only to some restricted extent. It is called selectively permeable because it allows only selected substances to enter and leave the cell. For the examples of substances may be oxygen, carbon dioxide, and water. It prevents what it means plasma membrane prevents the movement of other substances. It does not allow cytoplasm. This cytoplasm is not visible here. All this is cytoplasm in here inside the cell. So cell membrane does not allow the cytoplasm to move out of the cell. In animals, plasma membrane is the outer covering of the cell, while in plants and prokaryotes, it is usually covered by a cell wall, which is the outer covering outside the cell membrane. If now the question arises, what will happen if this plasma membrane breaks down or ruptures? What will happen? There will be free movement of all these substances in and out of the cell. The contents of the cell, that is the cell organelles, whether these are mitochondria, Golgi apparatus, nucleus, or ribosomes, all the contents of the cell, and also the liquid which is present inside the cell, that is the cytoplasm, in which all these cell organelles travel or move, will leak out, and the cell will not be able to perform basic functions. We can say that if this is the cell wall, a plasma membrane, and it breaks, all the parts which are present inside which will go out, all the material which may be present may move in, and the cell will not be able to perform the functions, and such a cell with broken down plasma membrane or ruptured down plasma membrane will die. What is plasma membrane? It is flexible. It is made up of protein and lipid molecules. However, we can observe the structure of the plasma membrane only through an electron microscope. We cannot see the structure of the plasma membrane with our naked eye. We will have to take the help of electron microscope. Hello students, myself is Parminder Singh from Prab Kirpa classes. Our today's topic is the fundamental unit of life, part 2. In part 1, we have already learnt who discovered cells, what are the three basic components of the cell theory, what is the difference between multicellular and unicellular organisms, why cell is called structural and functional unit of life. We have also learnt how division of labor is possible in multicellular organisms and why plasma membrane is called selectively permeable membrane and what happens if the plasma membrane ruptures or breaks down. We have also learnt about the process of diffusion and osmosis. We have also learnt the meaning of hypertonic, hypotonic and isotonic solution. In this course, we will discuss what is cell wall and whether the cell wall is found in animal cells. Next, what is meant by plasmolysis? Then, what is cytoplasm? Where is cytoplasm present? Then, what is meant by nucleolus? Then, if the organization of the cell is destroyed due to some chemical or physical influence, what will happen? Name the cell organs which are the sites of protein synthesis. Then which cell organelle is known as the powerhouse of the cell? Then what is meant by lysosomes? Whether lysosomes are present in plant cells? Why lysosomes are known as suicide bags of the cell? Then name of the two organelles that contain their own genetic material. Why plastids are called the kitchen of the cell. Then what are the various differences between animal cells and plant cells? Then what are the various differences between prokaryotic and eukaryotic cells? Then which type of cell division is required for growth and repair of the body and which type of cell division is involved in formation of gametes. In this course, we will learn about cell wall, cytoplasm, nucleus and ribosomes. First, cell wall. Plant cells, in addition to plasma membrane, have another rigid outer covering which is known as cell wall. This is picture of a plant cell in which you can see that this is the plasma membrane or cell membrane which is inside and this is the outer covering. This is known as cell wall. So you can see from this picture that this plant cell has two coverings. One is plasma membrane and the outer covering to the plasma membrane is known as cell wall. Plasma membrane is found both in the case of plant cell as well as in the case of animal cell, whereas cell wall is found only in the case of plant cells. Cell wall is found in bacterial and fungal cells. The cell wall lies outside the plasma membrane or the cell membrane. The cell wall is absent in animal cells. What is the function of the cell wall? Cell wall provides structural support as well as protection to the cell. Now the question arises, from which material cell wall is made up of? The plant cell wall is mainly composed of cellulose. The presence of the cell wall enables the cells of plants, fungi and bacteria to exist in hypotonic media without bursting. This is the picture of the hypotonic solution. This is the picture of hypertonic solution. If the plant cell is placed in hypertonic solution, you can see here that if this is the plant cell which has been placed in the hypertonic solution, what will happen? You can see that solutes are present in less quantity in the cell. whereas outside the cell, solutes are in dense quantity, more quantity. So what will happen? The water will leave from the cell to the outer, and only a few amount of water will come out. The net result will be that the more amount of water will leave the cell. And what will happen? Then the cell will shrink as the water will come out of the cell in more quantity. We call this contraction of the cell as plasmolysis. We have already learned about the hypotonic solution. That hypotonic solution is a solution in which the solutes are present in less quantity as compared to the another solution, that the solution inside the cell. When the plant cell has been placed in hypotonic solution, more water will come inside the cell than going out. So the net result will be that the plant cell will swell out, but the cell wall does not allow the cell to burst. It is only the cell wall which prevents the cell from bursting due to excessive water which has come inside the cell. Next, cytoplasm. You can see here that this is the cell, and the fluid which is present here all in the cell is cytoplasm, which is a fluid, gel-like fluid inside the cell. So cytoplasm is composed of salts and various organic molecules. In the cytoplasm, there are many specialized cell organelles. You can see here that this is the nucleus, this is the mitochondria, this is the Golgi body, these are the endoplasmic reticulum. There also lie ribosomes and lysosomes. These are mitochondria, Golgi body, ribosomes, lysosomes, endoplasmic reticulum. Each of these organelles perform a specific function for the cell. Cytoplasm is the ocean in which all the organelles float. You have seen that all these are the organelles, whether it is nucleus or mitochondria, Golgi body, these are endoplasmic reticulum, these are ribosomes. So all these are floating in the cytoplasm. Within the cytoplasm, materials move by diffusion. We have already learned about the process of diffusion in the part 1 of this course. Next, nucleus. In this picture, you can see that this is the nucleus. This is the control center of the cell. It is an oval shaped body. This is the oval shaped body which is floating in the cytoplasm. We have already told you that this is all cytoplasm in which all the organelles are floating. It is not possible to see the nucleus with the naked eye. To see the nucleus, we have to take the help of a microscope. Nucleus consists of nuclear membrane, that is outer covering, nucleoplasm, nucleolus, chromosomes. Nucleus is separated. This is the nucleus. This is separated from the cytoplasm by a membrane which is called nucleus membrane. It means that there is an outer covering outside the nucleus which separates the nucleus from the cytoplasm. Nuclear membrane keeps the other organelles and components of the cell outside. There are small pores in this nuclear membrane that allow selected materials to go in and out of the nucleus. With the help of microscope of higher magnification, we can see a smaller spherical body inside the nucleus. You can see here that this is nucleolus, a small spherical body which is present inside the nucleus is called nucleolus. In addition to nucleus contains thread-like structures which are known as chromosomes. What is the purpose of chromosomes? These carry genes and help in inheritance or transfer of characters from parents to the offspring. The chromosomes are rod-shaped structures and are visible only when the cell is about to divide. Next, in case of prokaryotes, you can see here that this is the picture of a prokaryote. This is the picture of eukaryotes. In this picture, you can see that the nucleus area is well defined, whereas in the case of prokaryotes, nuclear region is not well defined. It is poorly defined. In the case of prokaryotes, nuclear region is poorly defined due to absence of nuclear membrane and known as nucleoid, whereas in the case of eukaryotes, nuclear region is well defined and surrounded by a nuclear membrane. Now the next question before us is, if the organization, the cell is destroyed due to some physical or chemical influence, what will happen? We all know that as a building is made up of bricks, in the same way, all the living organisms are made up of small tiny units called cells. Cell is the structural and functional unit of all living organisms. If organization of the cell is destroyed due to some physical or chemical influence, all the activities performed by different parts of the cell, such as growth, reproduction of the cell, production of energy, removal of waste material from the cell, etc., will stop, and the cell will die, because there are many cell organelles which are present inside the cell which perform different type of functions, and if this organization of the cell is destroyed, then the cell will naturally die, because the functions performed by different cell organelles will not be performed. Now, what are ribosomes? The proteins are synthesized by ribosomes. Ribosomes are sites of protein synthesis. The ribosomes are very small and roughly spherical structures. You can see here that these are small spherical structures. These dots are small spherical structures. Ribosomes can be found floating within the cytoplasm, or they are attached to the rough endoplasmic reticulum. These are the rough endoplasmic reticulum, and ribosomes are attached to it. In this lecture, we will learn about mitochondria, Golgi apparatus, lysosomes, and plastids. First, mitochondria. In this picture, you can see that these are the mitochondria. Mitochondria all are small organelles inside the cells. You can see in this picture that the other organelles found inside the cell are nucleus, endoplasmic reticulum, Golgi bodies, ribosomes, etc. Mitochondria is present in both plant and animal cells. Mitochondria are known as the powerhouse of the cell. They are involved in releasing energy from food. Mitochondria stores and releases energy in the form of ATP. ATP is known as energy currency of the cell. Mitochondria uses oxygen and glucose to make OTP. Mitochondria move freely in the cytoplasm of the cell. You can say that mitochondria may move from here to here in the cytoplasm, from here to here, here to here. Mitochondria cannot survive outside of the cell. Mitochondria also make chemicals for other cell purposes such as breaking down waste products, responding to infections and injury, etc. Some organisms have a single large mitochondrion. Other organisms have thousands of mitochondria. Some organs like muscle, liver, kidney, brain contain higher number of mitochondria. There may be a question in the exam why mitochondria are strange cells. In what sense they are strange cells? So the answer will be the mitochondria are strange cells in the sense that they have their own DNA and ribosomes. So the mitochondria are able to make some of their own proteins. Now the next topic before us is Golgi apparatus. In this picture you can see here that this is the Golgi body or Golgi apparatus. Golgi apparatus is also known as Golgi complex, Golgi body, simply the Golgi. It is an organelle which is found in most eukaryotic cells. Golgi body was discovered in 1898 by Italian physician Camillo Golgi during an investigation of the nervous system. Now, what is the function of the Golgi body? Golgi body performs the function of modification, packaging, and transport of proteins and lipids, which are sent to different parts of the cell outside the cell. If there is no Golgi apparatus, then the packaging and transport of material to different parts of the cell and outside the cell will not happen. Then the other cells and organs in the body would not function properly without the products that the Golgi body normally sends. Now we will learn about lysosomes. Lysosomes is a membrane-bound organelle that contains powerful digestive enzymes. In this picture, you can see that these green color dots represent the lysosomes. Lysosomes are known to contain more than 60 different enzymes. This cell organelle is very acidic. Lysosomes are like the stomach of the cell. They digest dead or worn out or poorly working organelles. This is the function of the lysosomes. They also digest viruses or bacteria that enter the cell. They also remove the cell debris. What happens if the cell gets damaged beyond repair? If the cell gets damaged beyond repair, the lysosomes may burst and the digestive enzymes which are present in the lysosomes may eat up, digest other organelles of its own cell, which may lead to the death of the cell. So lysosomes are known as the suicide bags of the cell. So it means that if the cell is damaged from any part and it is not repairable by the internal system, then the lysosomes which are present inside the cell will eat all the other organelles which are present inside the cell, which will lead to the death of the cell. So due to this reason, the lysosomes are known as suicide bags of the cell. It is to be noted that lysosomes are absent in prokaryotic cells but present in all animal cells and in a few plant cells, that is fungi and yeast. Next, plastids. You may already know that plastids are not found in the case of animal cells. The plastids is a double membrane bound organelle. It is found in the cells of plants, algae, and other eukaryotic organisms. Like the mitochondria, the plastids also have their own DNA and ribosomes. The plastids provide color to the fruits and flowers. The plastids are involved in manufacture and storage of food. Plastids are called the kitchen of the cell. Why? Because plastids contain a pigment called chlorophyll, which plays an important role in the process of preparation of food by the plant cells called photosynthesis. It is because of the presence of the pigment called chlorophyll that the plastids are known as the kitchen of the cell. Because the chlorophyll plays an important role in photosynthesis. Now, there are three types of plastids. You can see in this picture chromoplast, chloroplast, and leucoplast. Leucoplasts are white colored or colorless plastids. They do not participate in photosynthesis. They are primarily organelles which store food such as starch, oils, and protein granules. Whereas chromoplasts are colored plastids other than chloroplast, usually containing yellow or orange color pigment. Now, chloroplasts, these are green colored plastids which help in the synthesis of food due to the presence of chlorophyll. Now, name the two organelles that contain their own genetic material. We have already learnt that mitochondria and the plastids are the two organelles that contain their own genetic material. Both these organelles have their own DNA and ribosomes. Now we will learn about endoplasmic reticulum, vacuoles, and cell division. First is endoplasmic reticulum. These two words consist of endoplasmic and reticulum. Endo stands for inside. Plasmic stands for cytoplasm and reticulum stands for network. It means that network inside cytoplasm. So collectively the endoplasmic reticulum is a large network of membrane bound tubes and sheets. In this picture you can see that these cream color and these stand for endoplasmic reticulum. The endoplasmic reticulum in essence is the transport system of the eukaryotic cell and has many other important functions such as protein folding. There are two types of endoplasmic reticulum. First is rough endoplasmic reticulum and the second is smooth endoplasmic reticulum. First we talk about rough endoplasmic reticulum. Rough endoplasmic reticulum looks rough under a microscope. Why it looks rough? Because it has ribosomes. You can see here that these red color dots represent the ribosomes. Because a rough endoplasmic reticulum has ribosomes are attached to the surface of the endoplasmic reticulum. The ribosomes are the sites of protein manufacture. The manufactured proteins are then sent to the various places in the cell depending on need using the endoplasmic reticulum. Now the smooth endoplasmic reticulum. The smooth endoplasmic reticulum has no ribosomes. It appears smooth. It helps in the manufacture of lipids. The smooth endoplasmic reticulum also plays a major role in detoxifying many poisons and drugs in the liver cells of the vertebrates. Now we talk about vacuoles. Vacuole is a membrane bound organelle. These are small organelles which are found in animal cells, and whereas in the plant cell the vacuole is of much higher size as compared to the animal cell. Vacuole may or may not be present in the case of animal cell, whereas a vacuole of a higher size is present in the case of plant cell. Vacuole is a membrane bound organelle which is present in plant and fungal cells and some protist animal and bacterial cells. The vacuole is filled with water, sugar, minerals and proteins. As already told, animal cells may have no or many small vacuoles. In this picture you can see that there are 3 vacuoles in the animal cell. Most mature plant cells have one large vacuole. The central vacuole of some plant cells may occupy 50 to 90% of the cell volume. What is the function of the vacuole? The main function of the vacuole is to handle waste products or store useful substances like water, proteins, etc. that the cell will need later on. In this picture, this is the vacuole which has been shown in the case of plant cell. In a single cell organism like amoeba, the food vacuole contains the food items that the amoeba has consumed. Now we discuss about cell division. The process by which the new cells are made is called cell division. Cell division is a process by which a mother cell divides into two or more daughter cells. When the cell divides, everything which is present inside the cell also divides, which means that the things like nucleus, chromosomes and cytoplasm also divide with the cell. Cell division is the process which makes growth and reproduction possible for any organism. There are two main types of cell division. First is mitosis and second is meiosis. First, mitosis. The process of mitosis has been shown here. Mitosis is the process of cell division by which most of the cells divide for growth. In the process of mitosis, each cell called the mother cell divides to form two identical daughter cells. Since that this is the mother cell and it divides into two daughter cells. The daughter cells have the same number of chromosomes as mother cell. Mitosis helps in the growth and repair of tissues in organisms. Now, meiosis. It is a special type of cell division of germ cells in sexually reproducing organisms. In meiosis, a cell divides twice. You can see here that a cell has divided first this, second time this. A cell divides twice to produce four. You can see that 1, 2, 3, 4. A cell divides twice to produce four new cells. This was the one cell and it has divided two times to produce four cells. The new cells have half the number of chromosomes than that of the mother cells. Now we will do the recap of what we have learned so far. Cells have a cell wall outside the cell membrane. This cell wall is not present in the case of animal cells. Nucleus is the control center of the cell. Ribosomes are the sites of protein synthesis. Lysosomes are known as suicide bags of the cell because whenever the cell is damaged beyond repair, the lysosomes eat up all the other cell organelles which are present inside the cell. Mitochondria are known as the powerhouses of the cell because they produce ATP, energy currency of the cell. Prokaryotic cells have no membrane bound organelles. Cells in the organisms divide for what purpose? For the growth of the body, for replacing dead cells, and for forming gametes for reproduction. Now the topic before us is movement of substances in and out of the cell. Some substances like carbon dioxide, which is a cellular waste, requires to be excreted out by the cell. Suppose this is the cell and carbon dioxide is in high concentration. Now we can see that we know that carbon dioxide cannot be seen for the purpose of example. We are taking this, assuming this, and suppose carbon dioxide is in high concentration inside the cell and outside the cell the concentration of carbon dioxide is less. As soon as there is difference in concentration of carbon dioxide inside and outside a cell, carbon dioxide moves out of the cell through the cell membrane. This is the cell membrane by the process of diffusion. That is from the area of high concentration to the area of low concentration. Similarly, you again assume that this is the cell and the level of oxygen inside the cell decreases as compared to the concentration of oxygen outside the cell. The oxygen enters the cell through cell membrane by the process of diffusion. Next, water moves in and out of the cell by the process of osmosis. These substances move in and out of the cell from the region of high concentration to the region of low concentration. It means that if the concentration is high in a particular area, it moves from that to the area where there is low concentration of the substance. Now, we will understand the difference between hypertonic, hypotonic and isotonic solution on the basis of concentration of solute in the solution. Solutions are of three types: hypertonic solution, hypotonic solution and isotonic solution. We will understand the meaning of solute and solvent in the coming slide. Using these terms, that is hypertonic, hypotonic, isotonic, always involves comparison between two different solutions. Let us assume otherwise. Suppose we say that he is taller. How much taller? He is taller than him. That is, we are making comparison of two persons. Next, if we say that he is more beautiful than her, we are making again comparison of two persons. In the same way, when we are talking about hypertonic solution or hypotonic solution, there must be the presence of some other solution with whom the comparison has to be made. Cells have a very limited range of conditions in which they can survive. It is not that if the conditions are too hot, too cold, too watery, too salty, too acidic or too basic, the cells cannot perform in all these conditions. They can function properly if there is the condition which is suitable to them. So we found out what happens when you place the cell in a solution which is more salty, that is hypertonic, much more watery, that is hypotonic, or has an equal salt concentration, that is isotonic. So we will try to find out the position of the plant cell when it is placed in hypertonic, hypotonic or isotonic solution. Now we will first understand the meaning of solvent and solute. Solutions are mixture of composed of solute and solvent. Suppose this is a jar, this is water. We have put some salt in it. So salt is a solute and water is solvent. A solvent is a substance that dissolves a solute resulting in a solution. A solvent is usually a liquid. Now what about solute? solute is a substance which is dissolved in another substance. In the example, you have seen that the salt has been dissolved in water. Water is a mixture, it is a solution, mixed solution which is composed of water and salt, in which salt is a solute and water is the solvent in which salt or solute has been mixed. Now we will define or understand the meaning of hypertonic, hypotonic and isotonic solution. We will try to understand the meaning with the help of pictures so that we can easily understand the definitions. First, hypertonic solution. In this picture, you can see that these dots are solutes. These are also solutes. So we are talking about hypertonic solution. The solution which is inside this black color covering is hypertonic solution. It has more solute in it as compared to another solution. That is, if we assume that this is the cell and the cell has less solute, whereas inside the cell solute is more. So the solution which is outside the cell is hypertonic solution. So next, hypotonic solution. This is the hypotonic solution in which the cell has been placed. You can see that solute is in low concentration as compared to the another solution, that is inside the solution, inside the cell solutes are in high concentration, whereas outside the cell solutes are in low concentration. So a hypotonic solution is the solution in which solute concentration is low as compared with another solution. Another solution means solution inside the cell. So now talk about the isotonic solution. If we assume that this is the cell and you find that the number of solutes inside the cell and the number of solutes outside the cell are equal. So outside the cell, this is the isotonic solution in which it has the same amount of solutes as compared with another solution, that is solution inside the cell. The number of solutes which are present in the solution outside the cell as well as within the cell are same. So whenever we are talking about hypertonic, hypotonic and isotonic solution, we are talking about the solution which is outside the cell. Now the next topic before us is what happens if the plant cell is placed in hypertonic solution, hypotonic solution and isotonic solution. These are three different situations. We will discuss the same one by one. First question before us is what is hypertonic solution and what will happen if a plant cell is placed in hypertonic solution, and what is the meaning of hemolysis? As already discussed, a hypertonic solution is the solution in which solute concentration is high compared to with another solution, that is solution inside the cell. All of us know that water molecules free to pass through the cell membrane in both the directions, that is water molecules can come in and go out of the cell membrane. If a living plant cell is placed in the hypertonic solutions, assume that this is the cell and it has been placed in the hypertonic solution, then what will happen? The liquid surrounding the cell has much higher concentration of solutes and the concentration of water is much lower compared to inside the cell. You can yourself see that solutes are less. Inside the cell, water is more. Whereas outside the cell, solutes are more, water concentration is less. Unfortunately, these solute particles which are present outside the cell membrane cannot pass through the membrane to reach equilibrium. So ultimately, the water molecules can pass through the cell membrane. So more water will leave the cell. You can see here that these are the pink color lines. More water is coming out of the cell. Only a small water is coming inside the cell. So what will be the net result? The cell will shrink because more water will go out of the cell and only small amount of water will come inside the cell, and the net result is more water goes out of the cell and the cell will shrink. We call this cell contraction plasmolysis. What this situation is called? This situation of cell contraction is known as plasmolysis. Now we take another example of hypertonic solutions. When compared to fresh water, sea water contains a higher concentration of salt particles. All of us know that sea water has much more salt particles. So the sea water is a hypertonic solution in which concentration of solute is on the higher side. Freshwater fish cannot survive in sea water because the cells would leak water into the surrounding salt water and they would quickly perish from dehydration. Suppose we assume that this is the cell of a fish and the fish is freshwater fish which has been placed in the sea water. Suppose this is the sea water and this is the cell of a freshwater fish, and what will happen? The water will come out of the cell of the fish into the sea water. Ultimately, the contraction of the cell of the fish will happen, and ultimately all the cells of the fish would leak water into the surrounding salt water, and ultimately what would happen to the fish? They would quickly perish from dehydration and ultimately it would lead to the death of the fish, as there would be dehydration, as more and more water will come out of the cells of the fish. Now the next question before us is hypotonic solution, and what would happen if the plant cell is placed in hypotonic solution? You can see from the picture that this is the hypotonic solution. This is in which solute concentration is low as compared with another solution. This solution is solution inside the cell. Inside the cell, solutes are in high concentration, whereas outside the cell, solute concentrations are in low concentration. When a plant cell is placed in a hypotonic solution, what will happen? Suppose this is the plant cell and it has been placed in the hypotonic solution. You can see here that solutes are in less concentration outside the cell, and whereas the plant cell has more concentration of solutes. Water is more outside the cell, whereas water is in less concentration inside the cell. So when a plant cell is placed in the hypotonic solution, water will come inside the cell by the process of osmosis. More water will come inside the cell. You can see here that rosy color pinpoints are there, 4, and only one arrow is going outside. It means that more water is coming inside the cell and only a small amount of water is going outside. So the net result is that more water enters the cell and the cell swells up, and coming of more water inside the cell builds up pressure against the cell wall. This is the cell wall. More pressure builds up against the cell wall because lot of water have come inside the cell. The cell wall then exerts an equal pressure against the swollen cell. In this way, the cell wall prevents the cell from bursting. We can say that as more and more water is coming inside the cell may burst, but the cell wall prevents the cell from bursting because it's also cell wall also exerts its pressure inside against the swollen cell. Now both tap and purified water are hypotonic. When an single animal cell is placed in a hypotonic solution, too much water rushes inside. Cell will grow in size first. It may be a question in the exam. What would happen if a red blood cell or a single animal cell is put in a hypotonic solution? The answer is that too much water will go inside the cell and the cell will grow in size and burst. We have in the previous slide we had seen that the cell wall prevented the cell from bursting. That was plant cell because it was having an extra covering, that is cell wall. But in the case of animal cell, cell wall is not there. Only there is plasma membrane or cell membrane. As the animal cells do not have cell wall, so when the water comes inside the cell in too much quantity, the cell grows in size and bursts, whereas in the case of plant cell, the cells do not burst. Now what is the meaning of isotonic solution, and what would happen if the plant cell is placed in isotonic solution? An isotonic solution is the solution which has the same amount of solutes as compared with another solution. In this picture you can see that the number of solutes outside the cell and inside the cell are equal. So the solution which is outside the cell is isotonic as it has the same amount of solutes. So what would happen if the plant cell is placed in isotonic solution? So the concentration of solutes inside and outside the cell is equal. The amount of water inside and outside the cell is also equal. So the amount of water going in the cell and coming out of the cell is equal. The net movement of water is zero because the solutes are equal. Equal quantity of solutes are present in and outside the cell. So the movement of water in net is zero, as the amount of water which is coming inside the cell is equal to the amount of water which is going out of the cell. You can see here that water is coming inside from two sides and water is also going outside. So the net result of movement of water is zero. In this case, what happens to the plant cell? The plant cell neither grows nor shrinks. It will stay the same size. Neither it will shrink nor it swell up. Here is another example of isotonic. Let us take 2 cups of tap water. One is big cup, another is small cup. In this big cup, we take 100 milliliter of water and we put in it 2 gram salt. After dissolving it, if we pour out some of this water into this, if we put out this solution into this, the concentration of solute in the solvent will remain the same. That is, the concentration of salt in the water will remain the same. So the solution which is present inside this small cup is also the same as compared with the solution which is present in the big cup. So the solution is isotonic, which has been placed in the smaller cup, because nothing has changed. The ratio of salt is 2 grams salt per 100 milliliter. The ratio will remain the same. Now we take another example of hypertonic and hypotonic solution. Suppose you like to drink cold milk having 1 spoon of sugar. When you do not put enough sugar in the milk, then it is hypotonic solution, because it has less concentration and it just tastes like cold milk without sugar. But when you put too much sugar in the milk, say 4 spoons sugar in your cold milk, then it is hypertonic, because the milk is now extra sweet. An example of isotonic would be perfect cold sweet milk that is not too sweet and is not too light. It is equal. Now we understand the meaning of what is the process of diffusion. Diffusion is the process of movement of particles, that is solutes, from a region of high concentration to a region of low concentration. In this picture, you can see that a purple dye has been put out in the water. Here the concentration of dye is more. It has slowly spread from the region of high concentration to the region of low concentration, and ultimately the dye color has spread, purple dye color has spread in the whole water. Diffusion is a passive process. It is not an active process. It is a passive process, which means that no energy is needed in this process. Breathing in oxygen and releasing carbon dioxide out of the body during respiration is an example of simple diffusion. Carbon dioxide and oxygen moves in and comes out of the cell by the process of diffusion. So the diffusion plays a very important role in the exchange of gases and water between the cell as well as the cell and its external environment. Now, what is osmosis and what is the significance of osmosis? The process of osmosis takes place in liquids. Osmosis is a type of diffusion. The spontaneous net movement of water or solvent molecules through a selectively permeable membrane from a region of high water concentration to a region of low water concentration till the equilibrium is reached is called osmosis. In this picture, you can see that this is the semi-permeable membrane. So in this part, you can see that solute particles are in less concentration and water is more, and this is a semi-permeable membrane. So slowly and slowly the water passes through this semi-permeable membrane as the water is in high concentration in this part of the jar, and and slowly it passes through the semi-permeable membrane till the equilibrium is reached. You can see here that the concentration of water in both sides is now equal. Equilibrium has reached finally. Osmosis is a passive transport. It does not require energy to be applied. Examples of osmosis in our daily life is plant cells soaking up water. Now, what is the significance of osmosis? Osmosis is very important for living organisms. Next, unicellular freshwater organisms and aquatic plants retain water through osmosis. Next, absorption of water by roots also occurs through osmosis. It means that the plants take up water through their roots by the process of osmosis. Next, movement of water in and out of the cell occurs through the process of osmosis. Now we will do recap of this course. The cells were discovered by Robert Hooke in 1665. Cell is the structural and functional unit of life. A unicellular organism, which is also known as single-celled organism, and it is an organism that consists of a single cell, whereas multicellular organisms are those organisms which have more than one cell, and the number of cells in multicellular organisms may range from a few to billions. The lifespan of a single-celled organism is short because the various life processes such as excretion, digestion are done by a single cell. So the lifespan of a single celled organism is short because of the load of work they have to perform, because load of work on the single cell in case of unicellular organism is very high. The lifespan of a multicellular organism is longer due to the reason that the different cells carry out specific functions. The cells of different living organisms may differ in shape and size. Next, shape and size of the cells are related to the specific function they perform. A cell consists of three parts: cell membrane, nucleus, as well as cytoplasm. Cell organelles are the membrane bound structures which are found in the cell, the various structures which are found in the cell. The plasma membrane is a biological membrane which surrounds the cytoplasm of a cell. Plasma membrane is also known as selectively permeable membrane because it allows only a selective material to go in and out of the cell. Next, on the basis of the concentration of solute in the cell, solutions are of three types: hypertonic solution, hypotonic solution, and isotonic solution. Now we will do important questions of this course. First question before us is, what is cell theory? The credit for developing the cell theory is usually given to two scientists. The name of scientists are Schwann and Schleiden. While Virchow contributed to the cell theory, he is not as credited for his stuff towards it. What are the main components of the cell theory? The three basic main components of the cell theory are: all living organisms are composed of one or more cells. The cell is the basic unit of life in all living organisms, and all cells arise from pre-existing cells. Now the question number 2 before us is, what is cell made up of? What is the structural organization of a cell? Or what are the three important features in almost every cell? So the cell has special components known as organelles. There are different components in the cell. If we study a cell under a microscope, we would come across three features in almost every cell. First is plasma membrane, which is the outer covering of the cell. Second feature is nucleus. And the third is the liquid or fluid material which is present inside the cell, in which the different type of structures which are found inside the cell are present. All the activities inside the cell and interactions of the cell with its environment are possible due to these features. So you can see that this is the plasma membrane, this is the nucleus, and the fluid which is present throughout the cell is cytoplasm. These are the three features which are present in almost every cell. Now we will make the comparison between plant cells and animal cells. These are the pictures of the animal cell, and this is the picture of the plant cell. Animal cells are generally small in size. Their shape is or irregular, whereas plant cells are larger in size. Their shape is rectangular. You can see that the plant cells are rectangular, fixed shape. In the case of animal cells, cell wall is not there. This is the plasma membrane, which is the outer covering. Whereas in the case of plant cells, this is the plasma membrane or the cell membrane which is on the inner side. Whereas on the outer side, there is cell wall, which is the outermost covering of the cell. In the case of animal cells, chloroplasts are not there, whereas as the plants are usually involved in photosynthesis, so the plant cells have chloroplasts. Lysosomes are present in the case of animal cells. These are the lysosomes. Whereas lysosomes are not present in the case of plant cells. An animal cell can often change its shape, whereas plant cells cannot change its shape. In the case of animal cells, no or small vacuoles are present, whereas in the case of cells, a mature plant cell contains a large central vacuole. This is the vacuole, large central vacuole. Now we will find out the difference between prokaryotic and eukaryotic cells. In the case of prokaryotic cells, the size is generally small, whereas eukaryotic cells are generally large in size. Prokaryotic cells are mostly unicellular, that is single-celled organisms. whereas eukaryotic cells are unicellular as well as multicellular. Eukaryotic cells are a primitive type of cell, whereas eukaryotic cells are an advanced cell evolved from prokaryotic cell. In the case of prokaryotic cells, nuclear region is poorly defined due to the absence of nuclear membrane and known as nucleoid. It means that proper nucleus is not present, as the outer covering, which is known as nuclear membrane, is not present in the case of prokaryotic cells, whereas in the case of eukaryotic cells, well developed nuclear region is present, which is surrounded by a nuclear membrane. In the case of prokaryotic cells, membrane bound cell organelles are absent, whereas in the case of eukaryotic cells, membrane bound cell organelles such as mitochondria, Endoplasmic Reticulum, Golgi apparatus are present. Next, in the case of prokaryotic cells, ribosomes are of 70S type, whereas in the case of eukaryotic cells, ribosomes are of 80S type. Prokaryotic cells are simple in structure, whereas eukaryotic cells are more complex in structure. The number of chromosomes in the case of prokaryotic cells are single, whereas in case of eukaryotic cells, the number of chromosomes are more than 1. In the case of prokaryotic cells, DNA nature is circular, whereas in the case of eukaryotic cells, DNA nature is linear. Prokaryotic cells are found in the case of bacteria, blue-green algae, mycoplasma, whereas eukaryotic cells are found in the case of plants, animals, and fungi. This point is also to be noted. On the basis of nucleus, cells are of two types: prokaryotic cells and eukaryotic cells. You can see here that this is the nucleate. Well-developed nuclear region is not present, whereas in the case of eukaryotes, well-developed nucleus is present. This is the nucleus. Membrane-bound nucleus. This is the. Now, what would happen to the life cycle of a cell if there was no Golgi apparatus? This is the Golgi apparatus. Golgi apparatus is also known as Golgi complex, Golgi body, or simply the Golgi. What is the function of the Golgi body? Golgi apparatus performs the function of modification, packaging, transport of proteins and lipids, which are sent to different parts of the cell and or outside the cell. If the Golgi apparatus is not there, then the packaging and transport of material to different parts in the cell or outside the cell will not happen. Other cells and the organs in the body could not function properly without the products that Golgi body normally sends, because the Golgi body is involved in sending different types of materials to different parts of the cell or outside the body. If transportation does not take place, so the other cells and the organs in the body would not be able to perform their functions properly. If the Golgi body does not send them the products, if the Golgi body is not there, then naturally it will not send the products which it used to send. Now, what would happen if the plasma membrane breaks down or ruptures? This is the plasma membrane, this is the covering. which the cell has in the case of animal cell. Plant cells also do have plasma membrane, but the plant cells also have an outer covering outside the plasma membrane, which is known as cell wall, but the cell wall is not present in the case of animal cells. So now we come to the main question: what happens if the plasma membrane ruptures, breaks down? Plasma membrane is also known as cell membrane or cytoplasmic membrane. Cytoplasm is present inside the plasma membrane. So the name of the outer covering is also given after that, cytoplasmic membrane. Plasma membrane surrounds the cytoplasm of the cell. Cytoplasm is present inside the whole of the cell, in which all these structures or the organelles are present. Plasma membrane separates the contents of the cell from the material outside the cell. This is the covering which keeps the outer material separate from the inside. If the plasma membrane ruptures or breaks down, there will be free movement. Suppose it breaks down, then there will be free movement of various substances in and out of the cell. Because it is broken, the many substances will come inside the cell and many substances will move out of the cell. The contents of the cell, that is these organelles which are present, vacuoles, mitochondria, nucleus, Golgi apparatus, all will leak out of the cell, and the cell will not be able to perform its basic functions. And such a cell which does not has its organelles to perform the various functions will ultimately die. Now, next question: which organelle is known as the powerhouse of the cell? Answer is mitochondria are known as the powerhouse of the cell. Mitochondria is present in the case of both plant cells and animal cells. They are small organelles which are present inside the cells. What is their function? They are involved in releasing energy from food. Mitochondria stores and releases energy in the form of ATP. ATP is also known as energy currency of the cell. Mitochondria move freely in the cytoplasm of the cell, which is the fluid present inside the cell. Mitochondria cannot survive outside the cell. Mitochondria use oxygen and glucose to make ATP, energy currency of the cell. Now, next question: where do lipids and proteins constituting the cell membrane get synthesized? The answer is lipids are synthesized in the SER, smooth endoplasmic reticulum, whereas proteins are synthesized in the RER, rough endoplasmic reticulum. Now, this is the normal question which we have been reading for quite a long time, and that pertains to amoeba. How does amoeba obtains its food? Amoeba is a single-celled microscopic organism which is found in pond water. It changes its shape and position constantly. It takes its food by extending like structures. You can see here that these are the arm-like structures which it has extended from any part of the body. It can extend arm-like structures from any part of the body. Pseudopodia means false feet. You can see here that this is the amoeba and there is some food particle which amoeba has seen, and it will extend its false arms, that is pseudopodia, and it will capture the same. Food vacuole will be formed. Ultimately, the amoeba will digest the same. The food trapped is gradually digested and absorbed. Amoebas eat algae, bacteria, and plant cells, etc. Phagocytosis is also known as cell eating. Phagocytosis is the process by which a cell uses its plasma membrane to engulf a large particle. Outer covering, this outer covering is the plasma membrane. Cell uses this plasma membrane to engulf the large particle. Small particles may be absorbed by the plasma membrane of the amoeba itself, whereas in the case of large particle, the amoeba has to extend its false feet. Now, what is the process of osmosis? Osmosis is a process which takes place in liquids. It is a type of diffusion, the spontaneous net movement of water or solvent through a selectively permeable membrane from a region of high water concentration to a region of low water concentration till the equilibrium is reached is called osmosis. Osmosis is a passive transport because it does not require energy to be applied. Example of osmosis in our daily life is plant cells soaking up water through roots. Now, next question, how does the movement of substances takes place into the cell? How do substances move out of the cell, or why diffusion is important? The movement of substance, the substance which may be liquid, solid or gas, from a region of its higher concentration to a region of its lower concentration is called diffusion. Carbon dioxide or oxygen moves in and out of the cell by the process of diffusion. So diffusion plays an important role in the exchange of gases and water between the cells as well as the cell and its external environment. Now next question, which type of cell division is required for growth and repair of body, and which type of cell division is involved in formation of gametes? There are two types of cell division. First is mitosis, another is meiosis. Mitosis is the type of cell division which is involved in growth and repair of the body, whereas another cell division is known as meiosis is this type of cell division which results in the formation of gametes. Next question, question 13. What is the meaning of ATP? ATP stands for adenosine triphosphate. It is a molecule which is found in the cells of living organisms. ATP is also known as secrecy of the cell. Mitochondria is the cell organelle which produces energy in the form of ATP molecules. Mitochondria supplies packs of energy in the form of packets of ATP. Whenever energy is required, the cell breaks down the ATP packet, which is a chemical reaction. In this reaction, energy is released. So the body of the organism uses this energy stored in the ATP for making new chemical compounds as well as for mechanical work. If you like our video, please like, share and subscribe our channel. Thank you.